Central dogma of immunity comprises of two pathways, exogenous and endogenous. Both the cell-mediated and humoral immune response are the result of these pathways. In an exogenous pathway, when an antigen enters the host body, it is taken up by the antigen-presenting cells, which can either be macrophages and dendritic cells. These cells degrade the antigen into smaller peptide fragments by phagocytosis, and present it on their surfaces through major histocompatibility complex class II. An activated T helper cell recognizes this presentation of antigen, resulting in its activation due to the binding of T cell receptor with antigen and CD4 receptor with MHC Class II. Activation of T helper cell can result in either the production of memory T helper cells, providing a much heightened immune response when the body encounter an exposure to same antigen, or the production of effector T helper cell which causes the release of cytokines. These cytokines can lead to the activation of B cells. When an inactivated B cell encounter an antigen or in the presence of cytokines, can result in the production of memory B cell for later exposure and plasma cells. These plasma cell produces antibodies which bind with the antigen resulting in its elimination from host body. Now let's talk about endogenous pathway. In this case, due to viral infection or presence of any oncogenes, cells become self-altered. These self-altered cells degrade these endogenous antigens, which can either be a virus or an oncoprotein, into smaller peptide fragments by the action of proteosomes, and is presented to the surface of these cells through major histocompatibility complex class I. Presentation of this endogenous antigen can result in the activation of cytotoxic T cells. An activated cytotoxic T cell gets activated when its T cell receptor binds with the antigen and CD8 receptor binds with MHC Class I respectively. Once activated, cytotoxic T cells can result in the production of memory cytotoxic T cells for later exposure and in the production of cytotoxic T lymphocytes. These cytotoxic T lymphocytes recognizes the self-altered cell, and upon binding with these self-altered T cells, it results in the degranulation of granules containing certain chemical toxins. These chemical toxins act on the self-altered cells, facilitating their removal from body.
Please like, share and subscribe, and don't forget to hit the bell icon for more videos.